Now it is my pleasure to introduce today's first plenary speaker, Dr. Michael Weiner. Dr. Michael Weiner is Professor Emeritus of Radiology and Biomedical Imaging, Medicine, Psychiatry, and Neurology at the University of California in San Francisco. He has been conducting research for more than 60 years and is Principal Investigator of the Alzheimer's Disease Neuroimaging Initiative, or ADNI, and the Brain Health Registry.org. In 1980, he was one of the first to perform nuclear magnetic resonance on an intact animal. He then pursued development of MRI and MRS as a clinical tool and established the Magnetic Resonance Imaging, or unit, which became the Center for Imaging of the Neurodegenerative Diseases at San Francisco Veterans Affairs Medical Center. Dr. Weiner has published over 950 peer-reviewed articles, has trained many dozens of postdoctoral fellows, and received numerous honors, including the Young Investigator Award of the American College of Cardiology in 1976. More recently, he received the Nancy and Ronald Reagan Award from the Alzheimer's Association, the Potemkin Prize from the American Academy of Neurology, a Doctor Honoris Causa degree from Paul Sabatier University, Toulouse, France, in 2019, and the Henry Wisniewski Lifetime Achievement Award in Alzheimer's disease research from the Alzheimer's Association in 2021. Please join me in welcoming Dr. Michael Weiner.
Well, we could just keep listening to Stevie Wonder. Thank you very much. We're going to talk about ADNI, my disclosures. Well, it's an honor to celebrate 20 years of the Alzheimer's Disease Neuroimaging Initiative, or ADNI. Where are we today, and how did we get here?
Major news in the fight against Alzheimer's disease.
Very important news for people suffering from Alzheimer's.
The first time yesterday, the FDA fully approved a drug that has been proven to slow the progression of symptoms.
Congratulations to Lily. So the story of ADNI over the last 20 years is really the story of the progress made in this entire field over the past 20 years. So how did it all begin? It began in 1999 with a paper in Nature by Dale Schenk at Athena Neuroscience, showing that if you inject mice with amyloid, they make antibodies against amyloid, and those antibodies will remove the amyloid plaques in those experimental animals. And that paper started what we now call immunotherapy. So 20 years ago, at that time, there were major limitations to do disease-modifying clinical trials. We didn't have a way to accurately diagnose Alzheimer's disease in living people. There were no treatments that slowed the progression of Alzheimer's disease. We had a need to plan what we call disease-modifying trials. We needed to detect and monitor the disease and the pathology. And at that time, the knowledge about biomarkers came from single-center studies, each focused on a single biomarker, and largely we had structural MRI, FDG PET, and cerebral spinal fluid measures. So there was also a need to standardize the measurements for multi-site longitudinal trials and compare the performance of the biomarkers and correlate them with cognitive decline. And finding a solution to these needs is what led to ADNI. So the overall vision and goals of ADNIs were at that time and continue to be, first, to validate biomarkers for Alzheimer's disease clinical trials. Secondly, and very importantly, to share all the data immediately without any embargo. And that was really new at the time. And finally, all of this was aimed to enable development of diagnostic tests and effective treatments. So a very big picture contribution of ADNI was that it was a new way of thinking about the interaction between academics, the National Institute of Aging at the NIH, biotech, and pharma, leading to the development of diagnostics and therapeutics. It really was a new model for the public-private partnership. And the model included, importantly, the National Institute of Aging, foundations like the Alzheimer's Association and ADDF, many academic sites, and many companies that were involved in developing diagnostics and treatment. So the big picture contribution really was widespread data sharing and a forum for exchange of information between academics, NIH, biotech, and pharma. And this model led to other studies, some of which are listed here, which are basically on the same model, public-private partnerships with data sharing. And it led to what we call the worldwide ADNI. There were ADNIs in many countries around the world, and the Alzheimer's Association had a worldwide ADNI program. And this slide shows the current members of the ADNI PPSB, the private partner scientific board. And now you're going to hear from some of the people who represent these companies. I can't hear.
Public and private partnership companies and government and academics in a way to collect data that was available to everyone. That seemed like a crazy dream, but it actually worked out.
It is the go-to data set and sample set for all kinds of scientific explorations that we do. It has also become a role model on several fronts.
ADNI has provided a common data source, a common language, a collaborative approach to research, to accelerate drug development, to understand the disease better, to develop biomarkers for diagnosis and tracking progression. It continues to drive the field and be the tip of the spear of Alzheimer's research.
So the ADNI, which began in 2004, is fundamentally a naturalistic study of AD progression. It simulates a clinical trial, but there's no treatment. We've enrolled over 2,500 study participants at 60 sites across the US and in Canada. And I really want to thank all of those participants and their families, because without you, we would never have the data, we would never have this project. All of our data is in a public database, USC Loney ADNI. You can go and enroll and download the data. There's no embargo. As soon as we get the data, it goes up on the website. And this has led to more than 6,000 publications. It's unbelievable. So what are the procedures? When ADNI got started, I was already a senior citizen, so I decided I was going to participate as a patient and have all of the procedures of ADNI, and I've been doing it for the last 20 years. Again, thank you to the participants. It begins with a cognitive test battery, and you can see a younger version of me having my tests at UCSF. We collect blood. And we banked all the blood, froze samples, because we thought maybe someday there'd be blood tests, and for genetics, lumbar punctures to get cerebrospinal fluid, all kinds of MRI scans, which you'll hear about, all kinds of PET scans, FDG, amyloid and tau PET scans, and all the participants get all of this at every visit or every other year. So this describes the complex organization of this huge project, with the participants at the center, the clinical sites who take care of those participants around them, and then the various cores, which I'm going to describe. And we are, the oversight is by the NIH and our steering committee meeting, our scientific advisory board, and the private partner scientific board of the various companies, because we want their input. So I'm going to review briefly what all the different ADNI cores have been doing all these years. First is our administrative core in San Francisco. We basically oversee everything. We try to coordinate everything, and we watch over the money. And over the years, you can see that we've been following the project with financial management. We started referring participants from the Brain Health Registry in 2016. And more recently, in ADNI 4, the current ADNI, we are doing the digital marketing. The clinical core, which is directed by Paul Azen and Ron Peterson, manages all the clinical sites. It's established and maintained, collects all of the data that comes from the sites. And this shows the various enrollments in the phases of ADNIs of cognitively normal, people with mild cognitive impairment and dementia. Importantly, in 2010, there was characterization of pre-dementia Alzheimer's disease, leading to prevention trials that Risa will be talking about a little later. And now in ADNI 4, we're aiming for 50 to 60% of our new enrollments to be from underrepresented backgrounds, and we are providing optional amyloid PET disclosure. An important paper from Mike Donahue showed that cognitively normal people with brain amyloid had a more rapid cognitive decline than people without brain amyloid, and led to the development of the so-called PAC composite, which is used in prevention trials. And by the way, there are many other composite scores of different types that have been developed using ADNI data. The MRI core is led by Clifford Jack, and they've collected 45,000 MRI series from over 10,000 exams and developed standardized MRI protocols, which have been used in many other studies, including clinical trials, some of which are shown on the bottom. This shows the many accomplishments of the MRI core, beginning with 1.5 Tesla MRI back in 2004, moving up to 3T in 2009, and now we are doing defacing of all the images to protect the patient privacy in ADNI 4. The PET core, led by Bill Jagist and Susan Landau, has collected over 3,700 FDG PET scans, over 4,000 amyloid PET scans, and 1,500 tau PET scans. We began with a pilot study supported by the Alzheimer's Association of carbon-11 PIB, and then florbetapir was introduced in 2010, flutalsapir in 2016, a number of other tracers as well, and now we've added visual reads of the amyloid PET scans. These are some papers from Jagist and Landau showing the baseline florbetapir results. Importantly, a comparison of the amyloid PET to CSF. Not that many studies have been able to do that, but we collected a lot of CSF. And this study shows application of the Centiloid scale so that multiple tracers, the data from multiple tracers can be all read in the same kind of framework. And of course, tau PET correlates with cognition and predicts cognitive decline much better than amyloid PET. The biomarker core, led by Les Shaw and A.D. Lee, who are based here at the University of Pennsylvania, has collected 28,000 participant samples over the years, and we make those samples available to investigators who request the CSF and plasma for their own assays, and then they give the data back to us, and it all goes up on the Loney website. So this shows the progress of the biomarker core. They did the first round robin, which is a multiple assay comparison of CSF back in 2009. The data from the biomarker core led to the FDA approval of both the Fujirebio and the Roche CSF assays. And now there's FNIH head-to-head p-tau comparison studies and amyloid studies. So here's a paper showing the work they did with the Roche Alexis platform that validated the Roche CSF platform against amyloid PET, leading to FDA approval along with Swedish Biofinder. And these are two papers showing head-to-head comparisons for plasma 2/40 and p-tau to predict amyloid status. The genetics core, led by Andy Sakin and Twansik No, has been collecting DNA throughout the entire project. They've been doing ApoE genotyping from the beginning, GWAS, whole genome sequencing, telomeres, and recently they discovered a gene for tau. The neuropathology core was started by John Morris. Rick Perrin at WashU now heads the Neuropath core, which collects samples, brain specimens from autopsy. We've got samples now from over 220 patients. It's a uniform, comprehensive pathology analysis. And the real, this shows how the number of autopsy samples has grown over the years. Now they are digitizing all of the slides, and all of the digital slides will be available on the Loney website. But this is a really key slide of this talk. This is the results of the first 100 autopsies. And basically what it shows is that only 11% of the ADNI patients have pure amyloid tau and neurodegeneration. The majority, 65%, have amyloid pathology plus other co-pathologies. And you can see from the table on the left that about 50% have Lewy body synuclein pathology, 45% have TDP pathology, vascular pathology, et cetera. So mixed pathology is very important in Alzheimer's disease, and we should never forget that. It's not just amyloid and tau. Laurel Beckett and Danielle Harvey lead the BioStat core, which has provided study design, statistical methods, compared accuracy of predictors of progression, and provide a lot of support to our users. Importantly, the informatics core, led by Arthur Toga, has really altered the scientific data sharing landscape. We were the first to do widespread data sharing with no embargo. And this has led to, as I said, 6,000 publications. 45,000 investigators have registered on Loney to download ADNI data. This shows the many activities of this website, but down on the lower right, 397 million MRI and PET downloads from the Loney site over the last 20 years. It's just amazing. A big problem with ADNI and with clinical trials and with most clinical research is that the research is not truly generalizable to the broad population because of a lack of diversity. This slide shows the racial composition of ADNI 1, ADNI 2, and ADNI 3, and these are the whites, and these are the people of color, and this is the census. So clearly we were way over enrolling white people, and we were not doing a very good job with enrolling people of color. And to address that issue, we formed the Diversity Task Force during ADNI 3. Ozio Mancanco and Monica Rivera-Mint led that effort. They provided supplemental funding to select sites to bring in more underrepresented people. We developed a marketing campaign, and this was during COVID, just towards the end of COVID. The enrollment of underrepresented people went from 93 to 163. That is, we went from 16% to 25% of the whole of ADNI 3 cohort was underrepresented. So it was a big accomplishment. And this shows now how in ADNI 3, after the Diversity Task Force, how we increased our diversity compared to the census. But this is the real problem, not just for ADNI, it's the real problem for our field. If you look at the US population, the census on the left, people over age 65 years, 67% of the people in the United States over 65 have a high school education or less. In ADNI, it's 13%. In other words, we're enrolling a lot of highly educated people, and even though we've increased black and brown population, we're still bringing in PhDs and master's degrees. We're not really addressing the problem of getting people who have healthcare disparities, and that's something we'll talk about more later. So because of the success of Monica and Ozioma, we formed the Engagement Core, which focuses on this issue with a culturally informed, community engaged research approach, founded hub sites with boots on the ground recruiters, and providing telephone support, established a health equity scholars program for young investigators. And now Dugu Tosin at UCSF leads the project, which takes all of the data together, puts it all together, and harmonizes it, assesses the effects of risk factors. She's developing imaging biomarker signatures for the non-AD co-pathologies, a clinical trial simulation tool, and recently a couple of papers on the cerebrospinal fluid alpha-synuclein seed assay, which accounts for a significant fraction of cognitive decline. But there are other major accomplishments: the Jack curves, the diagnostic criteria, and the framework, and let's not forget the treatments. And so many of you know these Jack curves. Clifford Jack, this was inspired by biomarker data from ADNI and other studies. There have been tons of papers with Jack curves. Here's a couple of examples. Now we have Jack curves for synuclein and Jack curves for Huntington's disease. And also the diagnostic criteria developed by the NIA and the Alzheimer's Association, basically very biomarker-based, inspired by ADNI and the so-called ATN framework led by Cliff, essentially a biomarker classification. But there are other major contributions. The CSF diagnostics, Fujifilm and Roche, have FDA approval. The amyloid and tau PET scans, florbetapir and fluortaucipir, their data from ADNI was used for their FDA approval. The early trials were based on ADNI data. Some prevention trials are based on ADNI data. And of course, the successful trials of aducanumab, lecanemab, and donanemab were based to some extent on ADNI data. And if you don't take my word for it, listen to the companies who did it.
At Avid Radiopharmaceuticals, we collaborated closely with ADNI initially to understand how amyloid imaging could work to identify patients and track patients longitudinally over time in clinical trials. If not for that advance, I think we still wouldn't have effective amyloid lowering mechanisms. It's also enabled subsequent generations of biomarkers. If we didn't have amyloid imaging in ADNI, we wouldn't have been able to do the tau scans in ADNI. If we didn't have the amyloid and tau, we wouldn't have been able to develop the blood biomarkers. When the science started to break in amyloid imaging, then that unlocked all of these subsequent advances that ultimately led to clinical trials that have now shown effective therapies for Alzheimer's disease.
ADNI was a very important part of Fujirebio's journey to achieve the first FDA authorization for our CSF amyloid ratio test. Without ADNI, this would have set us back years.
The ADNI study, including the samples and the data that's publicly available, have been absolutely instrumental in Roche developing this assay, bringing it to the market and getting regulatory approval. Without ADNI, this would have been much, much harder, would have taken much longer, and would have cost a lot more money in order to get the assay approved.
ADNI was very important in terms of the successful development and approval of lecanemab, both in terms of the phase 2 proof of concept study and the phase 3 confirmatory study. By helping us understand the right patient population, the right dose, and the right outcomes to ensure that we had a successful clinical development program. This meant that our phase 3 study was optimally designed to confirm the efficacy and safety of lecanemab.
The clinical trials for donanemab built on data, including data from ADNI, used amyloid imaging to select patients for therapy, and used tau scans to stratify patients. That was based on data from ADNI. And we did something that I think was really unique and remarkable, which is we stopped treatment with the amyloid-lowering drug once the plaques were clear, because we knew about the very slow rate of accumulation or reaccumulation of amyloid plaques in patients. Everybody in the Alzheimer's field would agree that there's no chance that we could be where we are today with the development of advanced therapeutics, with the development of new biomarkers for diagnosing and tracking the disease. We wouldn't have been able to get where we are today if not for the data that was built on a foundation from ADNI.
Without ADNI, treatments would be delayed. Patients would have to wait further, not only for current treatments, but future treatments. And ADNI is the cornerstone of this noble endeavor.
But we don't rest on our laurels. There are limitations to ADNI. First of all, it simulates a clinical trial. It is not an epidemiologic study, and therefore, for all the reasons I've been telling you, it is not really truly generalizable, and we need to improve that. We exclude people with moderate to severe cerebral vascular disease. We exclude people with Lewy body disease and seizures and Parkinson's disease. It's focused on biomarkers for clinical trials. The findings are not generalizable. Also, the technology keeps changing, and the MRI scanners and the PET scanners are getting better. So there is a lack of harmonization because we keep changing the technology as time goes by, and we're working on that. The high patient burden prevents us from collecting even more data. We're always being asked, could you get this, could you get that? But we can't do everything. And our database on the website is massive, and some people find it confusing, but there's a new website that's up now that's much better. I want you to go to it, and if you don't like it, let me know. So, but what are going to be the clinical trials of the future, okay? There's going to be increased emphasis on prevention, and you're going to hear from Risa about that. We're definitely going to have to attack more than amyloid and tau. We need to use a real-world, community-engaged research approach and reduce the emphasis on visits to the centers and bring the study more to the patients and into their communities, reduce the needs for scans, use more at-home online assessments and blood collection, using access to the electronic health record for screening and monitoring. And then how can AI help us? Adaptive trial design, perhaps voice robots for recruitment and assessment. Our future goals are more data on plasma biomarkers, including comparisons of different methods, and we've got to get more biomarkers to measure all factors causing dementia, including synuclein and TDP-43. And we have to do a better job at generalizability to include more people with low education and low socioeconomic status living in deprived areas. This is the single biggest problem in our field. So how can ADNI help you? Well, you can join ADNI as a participant, and you can learn about Alzheimer's research. We can help your site better enroll participants, especially underrepresented people, with our community-engaged approach and digital marketing. The data is available on the website. All you have to do is go, get a username and password, and sign the data use agreement, and you can request our ADNI samples on request. So in summary, our major accomplishments are: we developed a new model for public-private partnerships with data sharing leading to more than 6,000 publications, FDA approval of florbetapir and flortaucipir, FDA approval of the Roche and Fujirebio CSF assays, and we facilitated the design of trials for lecanemab and donanemab leading to FDA and CMS approval. There's so many people to acknowledge, we can't name them all. So you're going to see them on this credit roll. But if you are a patient or a family member, stand up. We applaud you. If you're at a clinical site, stand up. We applaud you. If you've been downloading data, stand up. If you're part of a company that's been using ADNI, stand up. If you've written a paper with ADNI, stand up. If you love ADNI, stand up. We've got a lot more to do. Thank you very much.